Okay, it's a great pleasure being here, and I thought I would give a lecture about some of our recent work on early peopling of the Americas, early peopling of Europe, early peopling of Asia, and then also touching upon what kind of possible ecological impact could these early people have on the extinction of the so-called Ice Age Megafauna. It means the big animals that was roaming in Europe, in Asia, and in the Americas during the last glacial period. So, I will start because it's, as we just heard, the area where I'm working in is called ancient DNA. So I just want to give you a very brief introduction to the field. So it all started back in 1984, where a guy called Alan Wilson from Berkeley, he showed it was possible to retrieve DNA from the skin of the quagga. This is a quagga. It's a zebra-like animal that lived and got extinct in South Africa in the end of the 1800s. So it wasn't a very old sample, about 100 years old, but it was a very important discovery because it showed that in principle you can go back through time and then look directly at genetic changes instead of simply inferring what kind of changes might have happened in the past based on DNA from contemporary today's organisms. So, and most people at the time thought, you know, the DNA molecule is a very unstable molecule and it will basically degrade due to spontaneous chemical reactions shortly after death, so there will be no chance of getting something from the deeper past. So this was the work of Alan Wilson's group. At this time and many years forward, people was concentrating when they tried to retrieve DNA from ancient material of so-called mitochondrial DNA. So mitochondrial DNA is a small genome, about some 12,000 base pairs that is found in our cells, and it's inherited through the mother line. So you all carry the mitochondria of your mother. And they're lying inside mitochondria's organelles in the cell. And you have many thousands of mitochondrial organelles, and in each organelle there's many thousands of mitochondrial genomes. So therefore it has a fairly large chance of surviving. If you're interested in a specific part of DNA from the mitochondria, you have a fairly good chance of it surviving through the past. Compared to the nuclear DNA, nuclear genome is about 3 billion base pairs, so much bigger. Half of it is coming from your father, half of it from your mother. But there's normally only 2 copies, right? So if you're interested in a particular part, you know, it's very often it's degraded. But of course, so in many years, people was focusing on this to try to reconstruct how has the populations of animals and humans and so forth changed through time. But what you're really interested in, if you want to understand the past, is really the nuclear genome. Another problem that came forward in the field in the early years was the problem of contamination. Contamination with modern DNA. And this was particularly a problem when we talk about human contamination. Because in these ancient materials, the DNA is highly degraded. There's very little left, if anything, that you can retrieve. So it means that it's very easy, you know, that your reagents and so forth, your reactions are getting swapped by DNA from the environment. And of course, human DNA is a major problem because it's humans who have digged out the fossils, it's humans who are doing the analysis, it's humans that are doing the reagents. So there's human DNA everywhere. And how can you actually distinguish if you dig up, you know, an old Viking bone, for example, and you're doing this work in Norway or Sweden or Denmark? How do you know, that the DNA is actually from the Viking and not from one of the researchers who have done the reagents and so forth? So therefore, in many years, until basically 2010, the general notion was it's impossible to retrieve reliable ancient human DNA sequences because of contamination. And this is why the paper we did in 2010 is very important, because it showed 2 things. It showed it's actually possible not only to retrieve mitochondrial DNA, but most of the entire genome of an ancient human individual. And it also showed that if you have this amount of data available, you can actually find the contamination and you can quantify the contamination and you can take it into account in subsequent analysis. And it really opened the possibility for doing very complex studies of our past, of how the populations of modern humans have changed through time. And this is really the approach that we and many other groups have been using over the last few years to try to revisit our history using genomics, ancient genomics. So one of the areas that my group has been very interested in is early peopling of the Americas. And this is because North America and South America are believed to be the last continents that was occupied by humans. So if we can't figure out what was going on in the Americas, I think there's very little hope that we can figure out what's going on in the rest of the world. Nevertheless, over the years, many different theories has been proposed as to how was the Americas populated. There's everything, from kind of the school book version that people came from Siberia and back, close to 20,000 years ago, there was a land bridge between Siberia and Alaska. People moved in here into Alaska, but then they were stopped because at this time there was some huge ice caps lying across North America blocking the way. And it was only about 13,000 years ago that these ice caps melted back, creating an interior ice-free corridor, 1,500 kilometers in length, where people then, according to the kind of the school book version, could move quickly through the ice-free corridor, move through North America into South America, and on the way, they developed the so-called Clovis Technology, which is huge spare points. This happened about 13,000 years ago, and when they moved down with these huge spare points, they killed off all the big-bodied mammals, all the megafauna on the way, the mastodons, the mammoth, and so forth. Then other people have said, well, we believe that there have been humans in the Americas earlier than 13,000 years ago. And they came maybe bypassed these ice caps by following a coastal route along the west coast here. Then other people have said, well, it's not, it's kind of assumed maybe that it's Native American that are, the ancestors of these early people, but there's been groups claiming, no, it was not Native Americans that was first in the Americas. It was actually Europeans crossing the sea ice that went much further south and, going like Inuits and Eskimos, hunting seal, etc., getting into North America, developing the Clovis technology, and then was later killed off or replaced, as we call it, by Native American ancestors coming in from Siberia. Still others have claimed that the first humans in the Americas is people related to Australians and Papuans, etc. Also, when we get into the Arctic part of North America, there's been large discussions how many waves of human came in there. We know archaeologically that there was humans there around 5,000 to 6,000 years ago, but were these people the ancestors of present-day Inuits occupying these areas, or were there other people living in there? There's even been suggested at some point Native Americans went up north, occupying Greenland and northern Canada. So if we start with the Arctic, my group, we collected almost all the ancient human remains that exist from the very early human remains from the Arctic, from Greenland and Canada and Alaska. We also collected early remains from Siberia. We genome sequenced these. We also sequenced the genomes of Native Americans and present-day Inuits. And then we analyzed this data, and it became very clear that there is two independent migrations of people into the New World Arctic. So 5,000 to 6,000 years ago, we have a group of people entering Alaska from Siberia, spreading across Canada into Greenland. And this group of so-called Paleo-Eskimos are surviving until just 700 years ago. So it's a whole population very recently that went extinct. And these guys, they are amazing in the sense that most populations contain several individual mitochondrial DNAs, from the mother line. These guys, we have only found one single mitochondria. So it's almost like, one or two women and a few men kind of crossed over here and then spread across the whole Arctic, northern Arctic. And strangely, when we start sequencing them, we could see that they were heavily inbred. And we could also see that they haven't had much exchange, genetic exchange with Native Americans, even though they must have met them to the south. So they seem to have been keeping for themselves, to a large extent, And because there's probably so few people coming in there, then after several thousand years, when you meet somebody, it's actually a close relative, right? Even if you are marrying, outside. So they become more and more inbred. Then a couple of thousand years ago, there's another group of people coming in from Siberia. And these are the ancestors of present-day Inuits. They're situating themselves in Alaska, developing a very sophisticated marine hunting technology for whaling and so forth. And then as soon as that package is developed, they're spreading very fast across the Arctic, all the way into Greenland. And as these people are spreading, the Paleo-Eskimos are going extinct. And we don't know whether they're killed off by the Inuits or whether it's diseases. That's something we're investigating at the moment. When we get to the lower parts, North America, how did it then happen? Well, as I said, a very crucial part of, you can say, of the debate is the so-called Clovis technology. That is, you can say, the earliest very widespread technology you find in the Americas, starting around 13,000 years ago. And as I also said, there has been people arguing that Clovis is actually Europeans crossing the Atlantic, the sea ice. And the reason being that Clovis technology, according to some archaeologists, looked very similar to the so-called Solutrean stone technology that you find in France and Spain around the same time. So, in other words, they're using the stone tools to say, well, this is a proxy for how people have been moving and how they were actually connected to each other. Strangely enough, you find Clovis technology all over North America, but there's only one single skeleton ever found in direct association with these Clovis tools. And this skeleton is found in Montana. And really strange, in America, at least in Montana, it doesn't matter whether it's a piece of gold or it's a skeleton you find. Then if it's found on private land, you own it. So this skeleton had been lying since the 1960s in a bank box in a farm in Montana. And then, I asked permission to go and do DNA studies, and many people had done this before, and they got no. But, I sent them the first ancient genome paper, and the daughter there, she's a molecular biologist, the daughter of the farmer who found it. And she said, well, I think you guys might be the right people, but I have one request, and I want to participate myself in the beginning of the study. And I said, yeah, come to Denmark. And she said, well, I have a small child. I said, well, bring the child. She said, who should take care of the child while I'm doing the work? Well, bring the bloody nanny as long as you're bringing a bone of that skeleton. And she did. And we sequenced the genome. And what became very clear is that this individual is genetically 100% Native American. Nothing European, nothing other Asian. It's a Native American, genetically speaking. And not only a Native American, this individual's, the group that this individual belonged to is the direct ancestor of basically probably most, if not all, of the people, native groups that are today living in Mesoamerica and South America. So it's really, you can say, a missing link, if you want. I always learned in school, it's impossible to find a missing link by chance, but, well, it's pretty close, I would say, to that. So it's also very clear, right, that these inferences, which is very often done by archaeologists, that, similarities in tools is also showing recently shared ancestry between groups, doesn't need to always be the case. I mean, obviously, here you talk about parallel evolution. I mean, I'm not an archaeologist, so I also wonder how many ways can you break a stone and get something useful out of it. But, so anyways, one thing I want to mention is that this kind of work with Native Americans is very sensitive. So there's there's historically very, very kind of tight relationship, very also to some extent hostile relationship between scientists and indigenous peoples in North America. So we are spending a lot of time going out to the tribes in the area and engaging with them. And their demand for this research was that the skeleton was reburied so the spirit could get rest. So a year after the publication, the skeleton was actually reburied in a large ceremony in Montana where tribes came from different places in the states and participated. Another skeleton, which probably is the most famous skeleton in the Americas, is called the Kenabeek Man. It's slightly younger. While the Clovis skeleton is 12,600 years, this is close to 9,000 years. The reason why it's very famous is that when it was discovered at the Colombian River, the first anthropologist looking at it, measuring the cranium and stuff, said, well, this has Caucasian features, implying, that this might be some kind of European. And then they got a C14 date, right, 9,000 years, and of course that kind of revived the whole Solutrean theory idea that, Europeans came in first. So, the tribes, there was 5 local Native American tribes that then claimed him as their ancestor and said, we want to rebury him, we want him back. And the US government, in the form of the Corps of Engineers, said, well, we want to give it back to you. But then a group of scientists sued the US government, said, well, we will not accept this because it's not a Native American. We don't believe it's a Native American. And there was a 10-year-long court case, and the scientists won. And the Native Americans were crying because it was their ancestor. They believed it's their ancestor. They tried to do DNA out on it, but they failed. Then we got a small piece of the leftover of the hand bone that was originally attempted to retrieve DNA from. And from there, we created the entire genome of Kenabeek Man. And what did it show? Well, it showed it's 100% genetically speaking Native American. So what the morphologists, the people doing cranial morphology had claimed was, well, he's either European-alike, but otherwise like Ainu, which is the indigenous peoples of Japan, right? So they published a book the same year as our DNA data came out, just a few months before, showing this feature of reconstruction of Kenabeek Man with a huge beard and everything. And of course, the natives were really upset. But now, because of the DNA result, the whole case has been opened again. And actually, the skeleton is most likely being returned to the Native Americans. And this was a big deal for them. You can see in the States, when we published these results, there was a huge press conference and stuff. Okay, now we know, it seems at least, based on the data available, it seems like Native Americans are deriving from the first people entering the Americas. But when are they entering the Americas, and what routes did they take? So one thing you can do with modern genomics, this is what we have done here, is you can say, a Native American genome, you can take the genome of its closest living relatives, which are Siberians, and then you can calculate when did these two populations split off, diversify from each other, and you can get a date of, let's say, 23,000 years. But that, of course, doesn't tell you whether that was the time where they were entering the Americas, right? They could just have, isolating themselves up in northern Siberia somewhere for 5,000 years before crossing into the Americas. So this is the problem with modern DNA, with DNA from living people. You can't place any events geographically. I mean, there you need the ancient DNA data. So if we start by looking then at the routes before we look at the time of entrance. So, as I told you about the interior ice-free corridor, right, that was opened, and you had this 1,500 kilometer walk through, with huge ice caps on each side that people believed the first humans entering Americas took. And if you have ever been in Greenland, close to, the edge of the ice cap, which is just really an unpleasant mud hole, right? You wonder how would it be to walk 1,500 kilometers, with 2 ice caps on each side. So even though it's physically open, you wonder, did they actually have anything to live from, right? Was there any animals to hunt? Was there any firewood? Surprisingly, people haven't, really done a lot of studies on that. So they just assumed, that people could move back and forth through this 1,500 kilometer corridor. So we decided to investigate this, and we used an approach that I developed during my PhD, namely the discovery that you can take ancient sediments, ancient soil, and then from the soil you can actually retrieve the DNA of the plants and the animals that were living in this place back in time. And the reason being that animals, through their feces, are releasing cells into the surroundings, and also through the urine are releasing cells. because of from the urine bladder, and that's getting out into the environment. Plants are constantly shedding, fine rootlets, and of course these things will degrade, right? They will disappear. But the DNA, because the DNA is electrically charged, and many particles, soil particles, are also electrically charged, then some of the DNA will bind to these soil particles, and those you can retrieve and then reconstruct it. So what we did here, you have the interior ice-free corridor. As you can see, this is the 2 ice caps on each side. Here we have Alaska. Here we have, lower 48 states. And then we went into, you can say, the last area that was deglaciated in the ice-free corridor, this bottleneck area. And then we drilled lake cores. So we drilled through the water column into the sediments of the lake, because the sediments are accumulating through time, right? So the deeper you go, the older is the sediments. The younger you go, the younger is it. Or the more the top you go, the younger is the sediments. So we could retrieve that, we could date it, and we could retrieve DNA to reconstruct how has this environment been. And we could see that although there was an opening 13,000 years ago, it was only by 12,600 years that you start really seeing a vegetation. You see a steppe environment, and as soon as we see the steppe environment, we also find the DNA from woolly mammoth, from bisons, and other steppe animals. Then after a few thousand years, you start seeing forest coming in there, like dispersed forest, and with that, other animals like elk getting in there. It's like red deer. And then after a few more thousand years, you get the boreal forest, that you see in northern North America today with spruce and so forth. And you also see a change in the animal life into moose. And it's amazing the details we can get out of these sediments. We can see when eagle gets in there. We can see when fish are getting in there. But the conclusion is it's very unlikely that the first humans entering the Americas could have taken this route, because it's first by 12,600 years that you can say that there's a viable system people could live from. And by that time, we know that at least for 1,000 years or so, there has been people south of the ice caps. It's more likely that they used this route later, when it became a real steppe environment. So in other words, it seems like more likely that they took a coastal route, although we haven't really investigated whether that was a viable route too. What about the timing? When did people get in there? Well, for many years, the general notion was it was first with Clovis people, okay, that humans got in there 13,000 years ago. And then we got involved with a guy called Dennis Jenkins who is doing excavations in southern central Oregon in some caves called the Paisley Caves. And there he's literally finding hundreds of ancient feces samples, dried in feces. And he came in contact with me and I said, well, could you please find out whether some of this feces is human? Because it looks pretty human to me, but I want to know. And to be honest, first I thought it wasn't very interesting. I thought whether somebody had taken a crab in a cave wouldn't really bring much important stuff with it. But I went and took samples, and they were lying in my freezer for 1.5 years. And then I got a young visiting student from Spain, and she said, Professor Willerslev, what should I do? What should I do? I don't have a project. And I said, look at the shit in the freezer. And that's basically what she did. And she came back and said, well, Professor Willerslev, I find human DNA that looks like Native American in some of these feces samples. And then when we looked in the stratigraphy, we could see that some of these feces samples were much older than Clovis, about 1,000 years older than the oldest evidence for Clovis. And of course, then Professor Willerslev got very interested. So we published first in 2008 a paper on this, and we were heavily attacked, because at that time, the general notion was Clovis is first, and people just wouldn't accept that in North America there was evidence of pre-Clovis occupation. And one of the criticisms that we got, I'm not an archaeologist, so I never really understood this, but was, why didn't you find any tools there? And I'm like, I don't bring my knife and fork with me on the toilet, but I mean, maybe some of the American archaeologists do that. But anyways, we got money because of this criticism to further investigate. And Dennis Jenkins was digging, and we took out feces samples just to confirm our previous result. But this time, he actually found 2 broken pieces of lithics, of stone tools, and this was in the Clovis layer, but the tools were not Clovis tools. It was something called the Western stem point tools. So Clovis first believers believed that everything comes from Clovis, right? Also the toolkits, also the people who are living in the Americas, and suddenly you're finding a complete different set of tools at the same time as Clovis. So it was a major blow to, you can say, the Clovis first hypothesis. So we know now that at least people have been there at least some 14,000 years, and this is obviously a minimum date. So where do Native Americans then come from? Well, I always learned in school that Native Americans is a group of East Asian people crossing the Bering Strait, getting into the Americas and becoming Native Americans. What is strange, however, is that although their closest living relatives are Siberians, they're actually quite distant, genetically distant from Siberians. And you have never found, you can say, a Native American in Europe or Asia. So it's kind of been a mystery. And solely by chance, really, we got our hands on a specimen here from southern Central Asia called Maltar. It's very close to Lake Baikal in Siberia, just on the border between Siberia and Mongolia. And this is an area that is today occupied by people of East Asian ancestry, different Mongoloid groups. That's what you call indigenous populations of this area today. But we sequenced the genome of this individual and got very surprised, because this individual is very closely related to present-day Northern Europeans, such as ourselves, but it's also very closely related to Native Americans, but it's not very closely related to East Asians that Native Americans should derive from. So what does this tell us? Well, it tells us that 20, 30,000 years ago, the distribution of people around the world, in some parts at least, was very different from today. And it also tells us that Native Americans are not simply, you can say, a group of East Asian people crossing the Bering Strait. In fact, all living Native Americans today, about 1/3 of their genome, which is a lot, is deriving from this Maltar population, or population very closely related to it. So it seems like Native Americans is probably derived from the meeting of at least 2 different groups of people. One group, which is this kind of Maltar European-like group, and then another Asian group, and you can say the child coming out of that meeting is probably Native Americans. What about if we go to Europe? Well, Europe has been occupied by modern humans, Homo sapiens, if you want, for about 45,000 years. But who are the first people living modern people in Europe? Is it people related to Europeans? Is it people related to other groups around the world? We actually had no idea. So we decided to sequence one of the earliest modern human individuals from Europe, the so-called Kościenki 14. It's an individual found very close to Moscow, dated to between 36 and 38,000 years old. And what we could see from this genome was a number of things. First of all, it's not a direct ancestor to present-day Europeans, but you can say among living people today, Europeans seems to be most closely related to Kościenki 14. We could also see, which was kind of surprising, that even 36,000 years ago, many of the major genetic components that you today find in present-day Europeans were already present in Kościenki 14. And then another group sequenced a genome of another individual from Siberia, Western Siberia, called Usteshim, which is 45,000 years old. And those 2 together could actually tell us something about when did Asians and Europeans diversify from each other? When did they split off from each other? Because Kościenki 14 doesn't contain any Asian ancestry, while Usteshim is equally distant to both Asians and Europeans. So in other words, and Usteshim is 45,000 years old. So in other words, it must have most likely have happened between 45 and 36,000 years ago that you had this diversification of Europeans and Asians. Okay, if we move up in time in Europe into what we call the Mesolithic, the late hunter-gatherers. Kostinki is an early hunter-gatherer, but the late hunter-gatherers here around some 8,000 years ago, they actually are quite similar, you can say, genetically to Kostinki. But one thing, we sequenced the genome of one of these late hunter-gatherers from Spain, from northern Spain, and one of the things that was very surprising was that this individual had already genetic adaptations towards, you can say, animal pathogens. So the general notion was that these resistances was something coming with agriculture. Because when agriculture comes, people are living very close with the animals, right? And then the idea was you're getting exposed to animal pathogens, bacteria and viruses. But this guy who is a clear hunter-gatherer has already a lot of these adaptations. So it really tells us that our conception of when things are happening are not always correct. We can also see that there's been a large discussion in Europe about, when we get agriculture, which happens about 10,000 years in southern Europe and 6,000 years in northern Europe, Scandinavia. How did that come about? How can it be that people stop shooting bow and arrow and killing animals and start milking cows and cutting hay? Was this due to, you can say, a spread of ideas? It means that the people were the same. They were staying in the same place, but they were just learning from neighbor to neighbor to neighbor how, it is to actually, start growing crops and stuff. Or was there actually a migration of people getting into Europe, bringing this knowledge with it? And together with my Swedish colleagues, we looked at some of this by looking at skeletons in southern Sweden, some of the late hunter-gatherers and some, you can say, very early farmers. And what you can see is that the hunter-gatherers, are very similar to people genetically today found in Eastern Europe, Russians, Finns, while the early farmers are very genetically close to Greeks. So in other words, if you go through, you can say, Europe, you can see that the more northeast you go, the more hunter-gatherer you become. The more south you go, the more farmer-like you become. And this suggests that there was indeed a migration of people from the south into the north. And we know now that these individuals who came, you can say, down from Anatolia in the Middle East, brought agriculture with them, had, you can say, a lot of interaction with the southern Europeans, but very little with the northern Europeans. So in other words, they had a lot of sex with the southern Europeans, very little with the northern Europeans, and when they got to Eastern Europe, well, they delivered, the knowledge, but didn't want to have any sex. And this is basically why, you can say northeastern Europeans are most hunter-gatherer-like in Europe. When we go further up in time, we get to the so-called Bronze Age period. It's between 5,000 and 2,000 years ago. And this is, again, a period of massive cultural changes, both in Europe and in Asia. And there's been a lot of discussion about what made this transformation. Was it migration of people? Was it spread of ideas? And archaeologists have been discussing this for decades. So we decided to undertake what was at the time the largest ancient genome study to date. So most genome studies has been 1 or 2 ancient genomes. This time we decided we wanted to do more than 100 ancient genomes. It was a massive undertaking, and we had to do a lot of methodological improvements to get this to work. But we collected Bronze Age samples across Europe into, Western Asia and all the way into Central Asia, close to Mongolia and those parts. Sequenced the genomes. And what we can see is, well, the Bronze Age was indeed a period of very significant movements of people. So if you go to early Bronze Age, 5,000 years ago, we can see that there's a spread of the so-called Yamnaya people from Eastern Europe into Northwestern Europe. Here they are meeting, the farmers, the early farmers living in Northern Europe, also in Scandinavia, and they are interacting with them. They are intermixing with them. At the same time, some of these Yamnaya people are crossing the Urals and going all the way into Central Asia. And here they seem to mainly be replacing the hunter-gatherers that were living there, like this Maltar boy, similar to that, at least locally. And this very early spread into the northwest and into the east, it's interesting because linguistics people that are dealing with languages and the development of languages, they have predicted, some of them have predicted that you had to have such a migration going on from the Yamnaya with the early spread of the Indo-European languages. So the Indo-European languages is what we speak today, and many other people speak today. So in other words, the Yamnaya people might very well have brought the language with them, at least the early version of it, that we're speaking today. Later in the Bronze Age, we actually see a migration going from Ural Mountains and Europe into Central Asia. This time it's the Santasta culture. And this is a warrior culture quickly moving all the way into Central Asia, replacing the Yamnaya people. And then in the end of the Bronze Age, in here in Central Asia, we start seeing people, various cultural groups of East Asian ancestry getting in there, mixing with the Santasta people. And it's very interesting in the beginning, when you look at these people, you take one skeleton, it's a European, another, it's an Asian. But with time, the Asians are becoming more and more dominant, and the Europeans are basically outbred. And today, in those areas of the world of Central Asia, you see people which is genetically almost complete Asians, but with a slight signature of Europeans in them. And most people thought, well, this is just the Russians or whatever, during the Soviet Union getting down there. But in fact, no, it's actually all the way back to the Bronze Age period. And what is also interesting is that these events during the Bronze Age has to a large extent shaped the genetic variation, the genetic diversity that we see in Europe and Asia today. So if you look here, all the gray spots represent the genetic diversity we see in Europe today. If you look at the late hunter-gatherers, you can see the genetic diversity is falling outside present-day diversity. When you go to the Neolithic farmers, it's also falling outside the genetic diversity. But then when we get to the Bronze Age, the genetic diversity that separates, for example, Southern Europe, Northern Europe, Eastern Europe, and so forth, are really start getting created. And the same for Asia, in the early hunter-gatherers, it's falling outside contemporary genetic diversity. Then we get to the middle Bronze Age with the Yamnaya expansion, it start getting created. And then with the Santasta expansion, and the Asians getting in there, we really start forming the genetic diversity that we see today. So in other words, a lot of the genetic differences that you see among people today in Europe and also in Central Asia is something that happened, over only the last few thousand years, right? It's very late in our history. So we can also start looking at phenotypic traits because we have so many samples. So for example, lactose tolerance. Lactose tolerance is the ability as adults to break down milk sugar. In other words, you can take, raw milk and you can drink it and you're not getting sick. This is an ability as adult which is very rare worldwide, but very common in Northern Europe. Some 70-80% of the population can do this in Northern Europe, while in other parts of the world, if they drink, raw milk, they get sick. Okay, we can of course all do it as children, but as adults. And so this is genetically based. And most people believed, well, this ability must have come during the farming, when the early farmers are getting into Europe, because then when people are getting a cow, and they are starving, the people who had to kill the cow and eat it can maybe survive, 1 month. People who can milk it and drink the milk are instead surviving half a year, right? And there will be a selection, and therefore it will come in. But what we can see is that during that period, and also in the Bronze Age, this genetic ability is super rare in Northern Europe. So in other words, this genetic ability to break down milk sugar as adults is something that first became abundant within the last 2,000 years. And what we can also see is that most likely it came with the Yamnaya people, because the Yamnaya has a higher frequency than in the Europeans. So they might very well, when they mixed up, with the Neolithic farmers, the early farmers, they have brought this genetic ability. And then, it over the last 2,000 years has becoming highly frequent. You can also start looking at phenotypic traits. So if you look at people, you know, the late hunter-gatherers in Europe, they would probably look pretty much like this guy here, very dark skin, blue-gray eyes, fairly short. It's with the Yamnaya people, you're getting people more like this, like Bloom here, with the very pale skin, the brown eyes, and tall people. So this is the Anaya Signatures. So it's very late again in our history that some of these traits, you can say, which is we consider typical Northern Europeans, are actually coming about. So what is creating this extreme dynamics in the Bronze Age? One group, you know, replacing the other. Yamnaya replacing the hunter-gatherers, locally at least. The Sentasta locally replacing the Yamnaya and so forth. So we were speculating about this, and first we thought, could it be something to do with climatic changes, but we couldn't really see any major climatic changes happening through that period. And then one of the archaeologists said, what about diseases? And the only disease we have ever heard about is really plague, because we are not medical people. So we have heard about plague, right? The Black Death. But the problem was, of course, that the Black Death happened, you know, 3,000 years later. But we decided to go and say, well, we give it a shot. We'll look, you know, for pestis, the bacteria causing the Black Death, in some of these skeleton remains. And this is one of the rare examples where you can be very lucky in science. Normally you're not. But here we were extremely lucky. So basically out of our 100 individuals, 8 of the individuals had the pestis bacteria. And we could sequence the whole genome of the pestis bacteria. We could sequence the plasmids. And we could look at it, how does it fit into, you can say, the phylogeny of present-day plague. And what we can see is that not only has plague been widely distributed during the Bronze Age, which was by itself a major surprise. It's before real cities was established and so forth. But we could also see, get a notion of how the disease has developed. Because in early Bronze Age material, it's the plague version we find that can only be transmitted from human to human by saliva. So you have to be in, you can say, close contact. It's super fatal. I think there's 100% fatality if you're not treated within 48 hours. But most people thought this was the derived form, the later form. But in fact, when we get to the end of the Bronze Age, then when we see this is the form that you see in the medieval times where the plague bacteria can be transmitted through fleas. So it can survive in the flea gut and then it can be spread through the fleas. So it really opens a possibility to study, you know, disease distribution, disease evolution, and now, of course, we're looking at all kinds of pathogens in our samples because obviously they don't only have plague, they have all kinds of nasty stuff back then so that you can explore. Okay, I'm not sure how I'm doing on time here, but otherwise you just have to stop me. But now I wanted to find, is it getting, okay, it's getting real close. Do you want to hear about the extinction of the Ice Age fauna? Yeah, okay. Well, at least some say yes. Okay, so one, you know, it's very heavily discussed. When did humans start having a really large-scale impact on the environment? Kind of a, you know, on a continental scale. And one of the earliest suggestions for this is the extinction of the so-called Ice Age Megafauna, right? So if you go back 10, 12,000 years in time, you would see an environment very different from today. And what made it very different was that you had a huge diversity of big-bodied mammals. You had elephants in the form of woolly mammoth, you had a woolly rhinoceros, you had lions, you had hyenas, something very similar to what you see on the African savanna today, literally running around in the snow. So, but for unknown reasons, 2/3 of these animals died out in the end of the last Ice Age, around 10, 12,000 years ago. And of course, this has been a major puzzle. What caused this extinction? And some people have argued, well, this was humans. Humans came out of Africa, we spread to the rest of the world. Up north there, we met these animals. They had never seen humans before, and we killed them one by one, and it happened really quickly, within 1,000 years of first contact. So you talk about extinction by blitzkrieg. Others have said it's ridiculous to think that a few humans with a primitive or simple technology could take out all these animals. It must be due to climatic changes. We know that the climate became warmer and more humid in the end of the last ice age. And in regarding to these groups, they say, well, that must have taken out the animals. The critics here says, well, how can it be? What was so special about this last warming period? Because these guys have survived many warming periods before without going extinct. So we decided to look into this and did what I think is still the largest DNA study looking at the population dynamics of these Ice Age mammals. So we collected bones and teeth from Europe, Siberia, North America, sequencing DNA from 6 of these species, 3 that went extinct and 3 that survived: woolly rhinoceros, woolly mammoth, horse, reindeer, bison, and muskox. And to make a long story, and then what we also did was we did what is called climate niche reconstruction. So when you have the fossil, you know I'm here in Siberia, I know the age because I've dated it, then you can use climate records from some of these areas to basically reconstruct what would the area that this animal have occupied through time, how would that have been if it's only driven by climatic changes, okay? Then finally, we also looked into the archaeological record, finding out how many, where has humans been when, how many animals have they consumed, and so forth. And to make a long story short, what we can see is there's a clear correlation between how many animals, what we call effective population size, how many animals are there through time, and climatic changes. So it's very little doubt that climate seems to have a major impact on the population dynamics of these animals. That's one thing. The other thing is that a number of these animals, it's very hard to see humans having much impact because they're not overlapping in time and space. For example, the woolly, the muskox that today is found naturally in Canada and Greenland, but back then it was also in Europe and Asia, and then it got extinct. But at the time of extinction, the muskox is not overlapping with any humans. They are way too far north, you know, so it's pretty hard to see how humans could have impacted it. So in other words, the data here really suggests that climate is a major driver of these changes. In some cases, humans may have locally been important for taking some of these animal groups out, but as the main driver, it seems to be the climate. So what we decided to do then was to basically find out what about the climate is causing the extinction. Could it be through the vegetation? And what we did was we collected more than 200 permafrost samples across, this is North America, this is Siberia and into Europe, and basically taking DNA out of those samples and reconstructing the vegetation history over the last 50,000 years. And what we then also did was we got access to some of these mummies of woolly mammoth, woolly rhinoceros. You know, sometimes in Siberia you find them frozen, right, with meat and everything on and skin lying in the frozen ground. So we got access to the stomach content of these individuals. And then we took samples of the stomach content that we sequenced to find out what were they eating. So what can we see? Well, first of all, what we can see is that if you go back 50, 30,000 years ago, the diversity of plants in this area in the northern hemisphere was much larger than today. And it was dominated by forbs. Then when we get around 20,000 years ago, what is called the last glacial maximum, the coldest and driest stage of the last ice age, there we see a major drop in the diversity, but it's still dominated by these forbs. And then we get into the Holocene, the next warming period, some 10, 11,000 years ago, and maybe one would expect, I thought anyways, well, then we would just return to the previous vegetation when it was warm. But no, then we see a complete different vegetation, a very massive vegetation change. So what does this tell us? Well, it tells us that even if you're returning to the same climate, it doesn't necessarily mean that the vegetation will follow. And this, of course, is important in these days when we talk about global warming. So even if you get the global warming under control and the vegetation has changed, right, it doesn't mean that you're returning to something you knew before. And it also tells you that when these critics of the climate theory said, well, what was so special about the last warming period? They have survived many warming periods before. Well, that might be true, but the vegetation from those warming periods might very well have been different, right? So it's actually through the vegetation that it's happening. And then when we looked at the stomach content, what we could see was that the dominant source of food for these guys were these forbs, these nutrient-rich forbs. So, and the nutrients forbs are really declining, you know, in the Holocene, I mean, the last 10 to 12,000 years. So, of course, a very, you can say, straightforward interpretation of this data would be, well, the climate is changing the vegetation, and the vegetation is really changed, is really what causing, you know, some of these animals to collapse. And then finally, because now I can see the chairman here is really getting, he wants to get this over with, I can see that. But, you know, just finally I will say, how far back in time can we go with all this? Well, we still have the record. We sequenced the genome of a 700,000-year-old horse from up from Alaska. And such an old horse, you can use it for many things, but one thing you can use it for is actually was a really important, you can say, issue in conservation biology. Namely, is that do there still exist any true wild horses? So all the horses we see today, you know, in Sweden, in Australia, America, in most parts of Asia and so forth, is really, even if they're wild horses, it's actually domestic horses that have gone wild. So the only candidate for true wild horse that has always been wild is the so-called Przewalski horse from Mongolia. But that was on the brink of extinction in the 1940s. There was like 12 individuals left or something like that. And of course, those Przewalski horses has been surrounded by domestic horses for thousands of years. So the question was, is there any true-blooded Przewalski horses left, I mean, that we can put some conservation efforts into? And the problem was, you know, you don't know what, I mean, beside the Przewalski horse, you don't know what should a wild horse genome look like, because there is no other wild horse, right? But of course, our 700,000-year-old horse, I can guarantee you it has been wild. And so you could actually use it, right, to infer, well, is there actually full-blooded Przewalski? Yes, and there was, in fact, some individuals, and that's, of course, where we should put the conservation. So finally, I'll just say thank you very much, and sorry it took me a little bit longer than planned. And then I would say, of course, this work has been done by a number of young researchers in my group, and I just want to show you the faces of the people who put in all the work. Thank you very much.
So, thank you very much, Eske, for this absolutely fascinating lecture. Before we start with questions and things, I would like to introduce Ebba Fischer, who is the chairman of the Crafoord Foundation.
Okay, I'll stay.
Okej, och jag hörs ju. Ja, tack. Först, Eske, vill jag vet att du förstår svenska,
ja.
Så därför talar jag svenska. Först så vill jag tacka dig för att du kom hit. Det var jättespännande att lyssna. Och du pratade ju mycket om det som har varit. Här är ju ett stort gäng människor som tänker på vad som komma skall. Och det är mycket därför som vi har de här föreläsningarna, för att ni ska bli inspirerade av naturvetenskap och forskning. Och att överhuvudtaget plugga, tycker jag. Det känns extra kul att ni sitter i stora salen idag på AF. För att precis som Dan sa innan, så att forska är inte bara nyttigt, det är kul också. Och det är kul att vara student. Och just här kan man ha väldigt kul. Det vet flera av oss vuxna som har varit här förr. Vi har till att börja med ett diplom till dig, Eske.
Mange tak.
Ja.
Mange tak. Tak.
Sedan har jag även en annan present till dig från oss i Kraftska stiftelsen.
Okay.
Dan berättade innan att Kraftska stiftelsen arbetar ju med annat än att ge bidrag till forskning. Vi har nu senast gett ett stort bidrag till ett fotoprojekt som 2 killar från Lund, eller män får man ju säga, för ni är ju killar som sitter här. De här männen är lite äldre. De heter Albert Viking som är fotograf och Oskar Edlund som har skrivit texter. De har i 10 år fotograferat människor över hela världen, kända och inte så kända, och människor som jobbar med mänskliga rättigheter, om deras drömmar. Och det här projektet har nu utmynnat i en fotoutställning som finns på Fotografiska i Stockholm. Okay. Och här är boken med bilderna. Och jag ska säga till er alla som sitter här att i mars så kommer den här utställningen till Lunds konsthall. Och jag kan verkligen rekommendera er att gå och se den då. Så jag hoppas att ni gör det. Än en gång stort tack.
Ja, tusen tack. Mange tack. Tack.
Tack så mycket, Ebba. So we continue in English. It'll be a little bit easier for Eske to understand your questions. We now have roughly 15 minutes or so, I think, for your questions. I suppose you have a few. So just raise your hand if you want to ask a question. There is a brave person far down there.
So, is it possible to make a copy of a mammoth in the future? And how much of the ADNA from the mammoth are already found?
Mm-hmm. Okay, yeah, very good question. Did you all hear it? Okay. No? Okay, but she basically asked, is it possible to, make the genome of the mammoth, and how much has already been done? And I guess also what you imply in your question is whether it's possible to recreate, right, the mammoth. So the genome of the mammoth has basically already been sequenced. To the extent it's possible, even from a living person or animal, you cannot sequence the entire genome. There will be regions that, because of structure or repetitions, etc., is very hard to sequence. But the amount that can be sequenced is already sequenced. In terms of, you can say, recreating the mammoth, it's not really my area. It's kind of a different field, but I think that it's probably possible to introduce, some of the mutations or substitution, if you want, that is found, the differences, let's say, between a mammoth and a living elephant. Some of those genetic changes you could, in principle, I think, introduce into, a fertilized egg of an Indian elephant. And therefore, potentially get something out that, carries mammoth features, if you want. It's not a mammoth, but has maybe mammoth features. It's at the moment, to the best of my understanding, it's not possible to, you can say, clone a extinct animal. And one of the reasons is that even, when things are very well preserved, like in permafrost, for example, the DNA is still heavily degraded. It's still very fragmented and destroyed. And I think in order to be successful here, you would have something very close to a complete nucleus, to make that happen. So it might happen with time, but I don't think it's happening, tomorrow.
All right.
And if you prefer asking questions in Swedish, it's okay. I think I will understand most of it, as long as I can answer in English.
Yep. So Swedish, English, or Danish.
Yeah.
Just pick the language you like. So, are there any other questions? I'm sure you've prepared questions.
Don't be shy. There's one there.
Got one up here.
Yeah.
You said when you analyzed the DNA of people living in Asia, you came to the conclusion that they're genetically related to Asians and slightly European. What's the sample size that you used to come to that conclusion?
Well, it's only, of course, the sample size in, you're talking about the Malta individual, right? The 23,000-year-old individual from Siberia, I guess. Is that true, or is that correct?
Yeah.
Yeah. I mean, there it's only a single individual. We have now found other examples of it elsewhere. But, so it's pretty simple, you can say, how you do it, right? You are comparing the DNA and saying, if you compare the whole genome, or what we have of the genome, and say how much does it resemble different populations or different groups of people around the world, right? And then you can see it has a very close affinity to these Northern Europeans, and even closer to Native Americans, and less so to East Asian populations.
Okay. I think we had one up here. So.
When and why did you decide to become a scientist?
What, sorry?
When and why did you decide to become a scientist?
Oh, a scientist. Okay, yeah. Well, that's something that came to me fairly late in life. So I have you can say I have a past actually of being an adventurous and fur trapper in Siberia, so something quite different. So after I returned from fur trapping in 1904. Then I had already started on the biology study, at university, but of course I had many breaks because I was traveling instead. But then I got very tired of freezing and eating bad food and stuff like that. So I decided to basically make, make another living. So I didn't think I would be a scientist, but at least I decided to complete my study. So that I completed my study, and then when I got to the final project, the master project, which is in Denmark 1 year, I did DNA, I retrieved DNA from ice cores, and then I got so interested in it. I mean, what you have to bear in mind here, it's sometimes reading about what other people do is pretty boring. I think at least it's pretty boring, right? And it's also pretty boring doing experiments where thousands of students have done it before you, and you know exactly what the outcome will be, right? It can be useful, but it's pretty boring. But doing new discoveries, doing something people haven't done before, it's amazing. It's just amazing. And to be honest, it was also amazing, traveling around in Siberia, and it was really wild and unexplored and all this, but it's nothing compared to the feeling that you get when you are discovering something new. It's like a drug. It's amazing. Think about you are the person in the world that just know this, that has discovered this, who have changed people's view on something. And it's a very, you can say also the process itself it's very hard sometimes, but it's extreme satisfaction when you are getting, when you are finally getting there. So I have never in my life, when I did my master and did this project where, you can say where nobody had done it before on ice, I'd never tried anything so wonderful in my entire life. And then I became a scientist. So, yeah.
Yep, so in 20 years' time you will be asked the same question.
Yeah.
Yeah, we've got a question over there.
For the future of your scientific field, what are you most excited about? What are the possibilities?
I think, to be honest, I think there's a lot of possibilities. And, I think that the field has a great potential to look, for example, at how can it be some people are getting things like diabetes type 2, some groups around the world, while others are not. I think that there's a chance to understand, you can say, how did we domesticate plants, animals, how did diseases evolve? I mean, it's just an endless amount of possibilities, and this is the beauty, right? I mean, it's a matter of choosing rather than, you know, you are forced to go in a certain direction because there's nothing else new to do. I mean, there's so many possibilities. I mean, both with the sedimentary DNA, where you can reconstruct these. I mean, one of the things, for example, we're interested in the moment is to look at rice domestication, taking cores, and then basically retrieving the DNA of rice through time, right? And then you can get all the, you can say, all the substitutions, all the genetic changes through time. Some of them might even have been lost in present-day rice and could be important. And because it's a plant, right, you can actually gene-modify rice and then create, if you want, the samurai sushi or whatever you want, and, you know, see, I mean, basically get genetic traits that potentially could even be important today. You could discover from the past. And this is just one of many possibilities. So it's very hard to say. I mean, this is always the problem, choosing between the many possibilities, really. But yeah.
Oh. So we have time for, oh, we've got one over there.
Yeah, I don't know if this is a stupid question or not, but
no, I'm sure not.
If I understood it correctly, you could take an old mammoth DNA and recreate a new animal with some mammoth features, right?
I don't think anybody have done it, so maybe also for ethical reasons, I don't know, but I think in principle it could be possible, yeah.
Yeah, I just wondered what would happen if you could take an old human and do the same thing.
Well, yeah, I guess that's possible, but probably even more ethical problematic. I mean, if you recreated something Neanderthal-like, I mean, you wouldn't know, should you put this individual in zoological garden or should invite him or her for coffee? I mean, I'm not sure. You know, so I'm not sure we'll get there. Hopefully not, I think, for ethical reasons. Yeah.
So we've got time for, say, one more question. And over there, yes.
Hi. From what I understood is that Siberians are more related to Native Americans than Asians. Is that correct?
Yeah, I mean, then yes, that's correct. So they are more, let's say, they are more closely related to Northeastern Siberians are more closely related to Native Americans than are, for example, Han Chinese or other Asians. Yes, this is correct. Yeah.
And I have another question. Yeah. How long did this study or research take? From the moment you got the idea?
I mean, yeah, it's a good question. So it's being asked, how long time does it take to do these research projects? And it varies a lot. I mean, some projects have taken a year, but there's other projects that have taken maybe 4 or 5 years to conduct. So it varies a lot, you can say, based on the scale of the project, right? So the first ancient human genome we did in 1 year. We also did nothing else, and we were, you know, a number of people working very hard to get it done. And then, you know, you have a study like the Bronze Age study where we sequenced 100 individuals. That took a couple of years. But then there's the permafrost study, you know, where we took these samples across the northern hemisphere. I mean, there basically I started collecting the samples in 2003, right? And the paper came out, I think, in 2015 or 14 or something like that. But it was every time I went on an expedition, I had a drill and a freezer with me, you know, in the northern hemisphere, and then taking these core samples, right? Because I had this plan, you know, about doing something like that, you know, in the end. So I just started very early on. So it varies a lot, but at least you can say it's something you can see if you start also as a student or, you know, a student or a young researcher and you're getting involved. I mean, you will see it to the end, from the beginning to the end. So you get this feeling of actually have completed a study. And I must really recommend you. I mean, I hope also, I don't know if there's teachers here today, but I mean, one thing you should really try, I think, out there in the high schools is to try to do, I know it's not so easy, but try to do new experiments. I mean, experiments which are different from what you read in the book. I don't know whether you're doing this today, but when I was a student, you didn't do that, right? It was like, every time doing the same thing, and you could just take, the report from the previous year and you could see the result, and it's super boring. But I mean, doing something new, I mean, I'm sure that a number of you will be just like amazed about, and the thing is, in natural science, this is the beauty, right? In natural science, compared to humanities, you can actually you can see something very clear, something new, right? It's not like, oh yeah, on one side, on the other side, and blah, blah. I mean, you have a very clear result, and it's actually really easy to do something new. There's so much to be discovered, also with very simple experiments. It's a wonderful feeling, so I would really recommend it. Yeah.
Thank you.
Yeah.
Thank you very much, Eske. Time flies. You will have chance to talk to Eske in about 20 minutes in person over a cup of coffee, if you like. But before that, I'd like to introduce a panel of 3 biologists from Lund University. So, please. So we have Marie Dacke, some of you may have seen on TV. We have Anna Nordén, who's a PhD student at the department, and we have Tobias Uller. So these are some of our most prominent youngish people. They were young once,
They were.
like you. The idea now is that they have actually prepared a few questions and things to Eske, and so I think you can run a discussion with Eske on your own.
Okay, thank you. Well, first of all, we would like to say what a fantastic lecture.
Thank you.
But not only that, I also need to say what a fantastic audience.
Yep. Yeah.
It's not often that we get whistles and cheers and so on. So keep that enthusiasm for science, because it is fantastic. So, Eske, can I ask you, because you said towards the end of your talk here, you said that now you can go back 700,000 years in time. Will this be the same in 10 years, do you think? Is this like because the DNA can't last longer, or is it just a new technique?
No, I don't. I think it will. There's still some development to gain in terms of you can say retrieving DNA, which are very, very degraded. I mean, of course, if it's all into single nucleotides, base pairs, it will be pretty hard, right? But I mean, at the moment, you can say you need a certain, for example, you need a certain size in order to map those pieces uniquely to a reference genome, right? So what you do is you're basically taking the small pieces out, and then you are comparing it to an existing genome. So for the humans, there's a reference genome, for example, and then you're basically saying, okay, where is this part fitting? And it's quite amazing that with 35 base pairs, I mean 35 of these letters, you can map something, I think it's like 60 or 70% of the genome uniquely, right? Which is really good. But of course, if you could go beyond that fragment length, I mean, let's say go down to 20 base pairs, that would really enable you to retrieve genomes out of fossils that are very degraded or very old, because the relationship between the copy number, the number of copies, and the fragment size in such ancient remains, it's an exponential function. So it means a lot whether you're just getting, let's say 10 base pairs shorter, right? It really has a huge impact. So, but the problem is you cannot map it uniquely, so somehow you have to look, I mean, at the frequencies, let's say, in a population, and they use that to say something about the probability of it fitting one place over another, for example, right? But I think that would gain a lot, for example. Then, of course, there's also, in terms of the chemistry itself, I mean, you can really, of course, change that and modeling that to be better, more efficient in getting the DNA out. So there's ways for improvement still.
Okay.
Yeah.
So mapping things to a reference genome, that's basically what you do. So I'm thinking about this environmental DNA that you look at. What if you retrieve the environmental DNA and then you found that there's a lot of things, woolly mammoth and muskox and stuff, but what if you find something that you can't map to a reference genome?
Yes, but then we can't identify it. So it's all being really relying on having reference DNA data to compare it to. So obviously you're getting DNA out of something you don't know what was down there, right? So you have to somehow, rely on it being present in the database. So there's huge databases that are also growing exponentially in numbers, right? Where you have all the DNA that people have been sequencing over time. And so in some cases, for example, with mammals, a lot has been sequenced, right? And you have a very good opportunity to map some of these things. But for example, with certain plants or insects, there what happens is that you get a match, but it's not really good match. And that, of course, either you can say, well, I can only say it's this family of plant, for example, or insects, right? You get a good support for that. It's within that family, but you can't say what species we are talking about. And sometimes even with mammals this happens. So I remember for the first paper, we got out some mitochondrial DNA, which looked definitely like a muskox, but not really 100% a muskox. And that's probably some of the extinct muskox forms that we know were living in these areas, but hadn't been sequenced, right? So,
isn't that very frustrating for someone who wants to discover new things?
Of course, it's frustrating not to be able to say what it is, but on the other hand, I mean, those databases are growing so rapidly that what we could, I mean, when I started, right, I mean, it was a real challenge, I mean, with a lot of the identifications. But now, I mean, have been growing so rapidly that it's just becoming better and better. And this also means that you can take the data, after a few years, and then you can actually reanalyze it and get even more information, which is also nice and beautiful, I think.
Can I ask you a little bit about human evolution?
Yeah.
So we for a long time we heard that humans have stopped evolving.
Yeah.
On the other hand, we're now seeing with the sequence data that there's evidence for strong selection in not just with respect to lactose tolerance, but other things as well. What's your take on this?
Well, I don't think we have stopped evolving. I mean, also just from the simple, you can say, rational that, I mean, it might be, you can say, in our part of the world, right, that people are not dying from diseases and stuff. I mean, but there's certainly other places in the world where this is happening and where you can imagine, you can say, a selection going on, right, towards, to prevent a certain disease or whatever. So I don't think, I've heard this before, that people say now we're only culturally evolving, right, but there's no biological involvement. We're not biological evolving, but I don't think this is the case. The other thing is that because of the globalization we are seeing, of course, I mean, people are mating with each other. I mean, even at the Bronze Age, it was people were mating over long distances. We can see this. But now, of course, this is even, you can say the possibilities are endless of finding new partners around the world. And that also means you will get a lot of new recombination and stuff that will create new diversity and so forth. So no, I don't think so.
If you're looking back in the history, the time that you have worked on, are there any particular points in time that you think have been particularly important for human evolution?
I think that there's little doubt that you can say from the time of the early farmers, and we will see up to date. We don't know the whole story yet, but the change from hunter-gathering to farming and the Bronze Age, there's no doubt that has been extremely important periods in terms of creating, you can say, the distribution and the genetic diversity that we see in people today. And it has been, many archaeologists of, you can say, my generation and the previous generation was really of the belief that people were not moving very much, right? I mean, that all the cultural changes happened through spread of ideas, but people were staying put. And I would say one lesson we have learned from these studies is that people very early on moved great distances, right? And you can say the whole idea of people just staying in a local area and looking around and saying, well, I have never been outside, my local area, is probably a phenomena that has been introduced much later in our history. But when you go back, I mean, we also did the strontium on one of the Bronze Age women from Denmark called the Eske woman, which is a very famous mummy. So we could use the strontium to say how has she moved over her lifetime through the hair, right? And she was probably born in Germany, came to Denmark, went all the way to southern Germany, and went back to Denmark again and died, right? So it's over long distances these individuals have been moving, and they probably knew more about the world than we think. And also early on, with the spread, we're also doing a lot of work in Australia, but I didn't have time to cover that. But, some of this early spread of people after we left Africa, you have an amazing spread of people, the ancestors of Aboriginal Australians and Papuans, covering huge distances, right? Even crossing ocean, 50,000 years ago or more, to get into, Papua and Australia. So, I mean, one thing we have learned, I think, is the space that people have covered, the many times people have met each other and intermixed. So the whole idea of races and stuff, I mean, I think is completely outdated. And then, and also people's abilities. I mean, we always looked at these kind of early humans as being really simple and primitive and sitting, banging in a stone, right? And they don't understanding much of the world. And I think this is completely wrong. I mean, these people were really capable of a lot of what we are capable of today.
I have a question about mixing, because you talk a lot in your talk about something dying off or something being replaced. But can it also be that like the Neanderthals, for example, maybe they didn't die off, maybe they mixed and then they actually just continued?
Definitely. I mean, I think for most groups probably this is the case. I mean, you can say they're dying out as a group, but there's signatures of the genetic material still surviving in us. I mean, another example is this Maltar boy we found, the half Native American, half Northern European-like individual living around Lake Baikal, right? I mean, to the best of our knowledge, this group of people doesn't exist anymore. I mean, you don't have anybody, you can say, with that genome, if you want, or very close to that genome surviving, but fragments of that genome has survived in various populations around the world, right? So I think this is basically the case. And we are all, I mean, I think this is important, at least in Denmark these years, where, I mean, compared to Sweden, there's a lot of anti-migration and stuff like that. It's an important message that people have been, we have been formed by migration and admixtures. It's just how it is, right? It has formed our genetics, it has formed our culture. And, sometimes I'm even called into political parties, to give some background, right? Because most Danes, for example, has the notion, that we came from the Stone Age man, all the way up to present day. And then first in the 60s something happened, right? But of course these results here shows, no, I mean, we are a product in Europe, as well as other parts of the world, of migration and mixing of peoples. And this is also what has created a lot of our culture, our language, and so forth.
So to ask about some of our best friends, the dogs.
Yeah.
So it's not only humans they have done ancient DNA studies on, but also the dogs have been fairly heavily studied. And if I'm not wrong, they've recently shown that dogs don't only have one origin, but actually two, so they come from two lines. How can they, I mean, how do you say that? Why is it not three? Could it be three if we could go even further back? And why do you say it's two?
Well, I'm not saying that. because I don't believe in it.
Okay.
I actually believe that you cannot say it because it's very complicated with the dogs. I mean, I would say there's even some results now, I think, suggesting that at least one of the domestication is happening from a wolf, a group of wolves that is no longer around. I mean, there's no descendants from that, right? And so it's the domestication process and the number of domestications, it can be extremely hard because to deal with, especially just from contemporary DNA, from living dogs, because there's been so much breeding and there's been so, you can say the same is the case with horses. So at the center we have a person, Ludovico Lando, who's working a lot with the horse domestication. And the whole bloody variation has been lost, right, in contemporary horses. And it's probably the same with dogs. I mean, so it's first when you go back in time, you can actually really see in details, right, what's going on. So you need to do a lot of sampling, I mean, to get a notion of what is going on there, right? Because if you build a tree, as you know, right, I mean, and you're missing certain parts, the branches will just swap in, right? Instead, it should have been out here and so forth. So it's a very difficult question, I think, this about the number of domestication events. Yeah.
So is there a risk that this type of analysis is overinterpreted?
Yes.
So we read more into it?
Yes, I think so. And I think, like any other field, you can say, which is very young, what we will find out, and we're already starting finding out, of course, that the history is very complex, right? So when we started with one genome, we were sitting there, inferring the history from, one genome, and then people came here, and then they came there. And it was really first with the Bronze Age study where we did 100 genomes. We could see, there's a lot of variation within, for example, Europe, right? I mean, there's a lot of differences. And so you can say you can't just you have to be very careful from a single genome, from a single locality, really inferring that. But it also depends on where you are in time. So you can say at the moment, if you go back to the early hunter-gatherers or even the late hunter-gatherers, the genomes that has been done and more genomes being done actually suggest something very similar. I mean, very little structure. There's some, but it's very little, right? And then you get up to the Bronze Age and you see massive structure. So there you really need a large sample size to do these inferences, right? But of course, it's like with any other field that you just, you can say you have to, of course, be careful what you're doing and how you're interpreting, but it's also a learning experiment. So this is another important take-home message. In science, you have to make mistakes to become wiser. It's just how it is, right? If you got it all right from the beginning, well, then you could close your computer and go home, right? There's nothing to do. So this is part of the process. I mean, becoming clever, more and more clever on the world is actually to make mistakes. Good.
Tiden går alldeles för fort. Och vi ska ha lite grann tid för mingel där ni har chans att prata med Eske och med vår forskarpanel. Så jag tycker vi börjar med att ge vår forskarpanel en varm applåd. Och sen naturligtvis en alldeles särskilt varm applåd till Eske som har delat sin entusiasm och sin kunskap med oss.
Thank you so much for your time. Thank you.